And people will check with us like we are. Our division has, I just counted, 23 geriatricians and 8 PPs, psychologists, social workers, and then researchers.
I know.
Sarah Weary. A lot of Harry Burrow. Wendy Crow.
All right, I'll start off with introductions. Welcome. Happy December. Today is door decorating day. You probably knew, but we are excited to decorate doors. So first we'll do grand rounds and then division meeting. Welcome to those who are on Zoom. We are on Zoom. Jessica, is that true? We're on Zoom? Perfect. So just brief announcements. Next Journal Club is next Thursday. Dr. Garcia, Dr. Orris. Next Grand Rounds is December 18th, Dr. Michelle Fullard. Evaluations will come around. Those of you who are in Zoom, please do chat in. We'll have time for some Q&A at the end. And so let me get to introducing our speaker. I'm really excited to introduce Dr. Joe Hippensteel. He is a physician scientist in the newly named Division of Pulmonary Allergy and Critical Care Medicine. His research focuses on the role of endothelial injury in critical illness, which includes focus on sepsis and acute lung injury, with a particular interest on long-term outcomes in survivors of critical illness, those folks who then come back to us in clinic in our various settings as well. He received a NIA GEMSTAR in 2021, which focuses on understanding the interplay between older age, endothelial injury, and sepsis outcomes, and is a UW alum.
Yeah. Well, thank you for having me. I'm going to cover a fair amount of ground, I think, today. I'm going to give hopefully some, even if you don't pay attention to the second 2/3 of the talk, the first third hopefully is going to be interesting or enlightening, or at least reinforce what you already know. The second 2/3, which is going to be about my work, will be sort of a longer story about where the GEMSTAR has taken me and what I'm hoping to continue to do. And then sort of a smaller side project that I would love feedback on. And if anybody's interested in it, would love to talk more about it. So this is different than the title that I initially presented, but I will talk about endothelial injury too in the middle of this. But really want to start with just a broad strokes overview of the background of critical illness in older adults, and actually spent quite a bit of time. I haven't done this historically, but it was nice to spend the time the last few weeks sort of digging into what we know about critical illness outcomes in older adults. And so I'll start with that and talk about long-term outcomes for people who survive their critical illness. And then I'll dive into my work, which will be nitty-gritty, sort of basic translational science, but we'll try to walk you through and hold your hand and help you understand what I'm working on. All right, so the first thing, incidence and severity of critical illness in older adults. I did just a census in our ICUs on Tuesday to kind of see where we're at with people who are over 65. And of course, we all know that age itself is not the fixed number, but gives you a general sense of what our ICUs look like here at Colorado. I actually thought it was kind of interesting. Our surgical trauma ICU is 50% patients older than 65, but the range goes all the way up to 90 to 95, as we all know, in both our step down and ICUs. And this does not include step down, but about a third of our patients as of Tuesday are older. And the thing I'll say is, while this is not perfect, and I think we expect this in the hospitalized patient, only 15% of our population in Colorado is greater than or equal to 65. So suggesting there's this disproportionate impact on older adults of critical illness, which is borne out in literature. So I tried to dig through as much as I could with the highest quality data that I could find. And if we look at, for example, a sort of cross-sectional study of patients in ICUs, in one study, they found that 45% of patients within their ICU were greater than 65 years old, again, accounting for a population of 15% for that age range. If you look at much significantly older, greater than 85, representing 3% of the population, it accounts for about 10% of those in the ICU. So again, sort of a disproportionate burden in the ICU for our older adults. When you look at the causes for admission to an ICU, The majority, or not the majority, but about a quarter are related to either cardiothoracic surgery issues or cardiac ICU problems like MIs, heart failure exacerbations, and things like that. But another quarter is near and dear to my heart, which is respiratory causes like lung injury as well as sepsis. So accounts for about another quarter of those who are admitted. So about a quarter of those that I see have probably a third to 50% of the patients that I see have either respiratory failure or sepsis as their cause of admission to the ICU, which is why I focus on that in my clinical and scientific world. When you look at what people get when they're in the ICU, about 5 to 10% of patients will get hemodialysis. About 50% will be mechanically ventilated. And vasopressors are pretty common. So about a third of patients admitted to ICUs will end up on vasopressors, so will require blood pressure support. And if you look at the top procedures and this was back in 2008, there was a AHRQ review of sort of what procedures are done in patients who are older. And I think mechanical ventilation is actually the third most common procedure that those in the ICU undergo. And this does not include central lines, which is not sort of a procedure that is included in that bundle that they included in the top 20, but dialysis is in the top 10 for patients over 65. And when you get even older than that, sort of 80 and above, it's still within the top 10. So dialysis and mechanical ventilation are quite common. And so this is a big burden. You all see sort of the outcomes of this when you see them in the clinic, but not uncommon to have invasive procedures in these patients. This is seminal work by Mark Moss, who was, as probably most of you know, was our prior division head, who looked at this from about 2000 to 2005. And he showed that in sepsis specifically, so sort of subdividing this population, very similar to what we see in the ICU at large, even though older adults account for 15% of the population, nearly 2/3 of admissions for sepsis are in patients over the age of 65. So again, disproportionate impact on older adults. And I think why it's important to have geroscience sort of integrated with critical care and critical care outcomes. And ARDS is similar, although the data is not quite as strong, are not quite as strong. So this is the only study that I could find that truly estimated the incidence of ARDS in older adults. And it was done in Granada. And you'll see that there were 4 cases in those 15 to 29 during this period. There were 26 cases in those 60 to 74, which you'll see here on the right, and 12 cases in those greater than or equal to 75. And so this is really our sort of point estimate of incidence of ARDS in older adults, suggesting that the incidence does increase with age. But again, this is a single country and sort of a population-based study that may not be generalizable to the United States or anywhere else in the world, but it's what we have to sort of argue that this is important for ARDS incidence. The other thing I'll say is there's some really good work in the early 2000s looking at ARDS in trauma specifically, so sort of one subform of ARDS. And it seems like there may be actually a peak in ARDS incidence between about 60 and 70, which may sort of hearken to the importance of biology in this, that maybe there's sort of a peak of biological variables that actually lead to an increased risk of incidence of trauma. And this was sort of adjusted for lots of other factors. So suggests that there certainly is age-related effect on ARDS, both incidence and likely severity. Okay. So here's a nice review of mortality data for older adults with critical illness. And these were I only pulled out the multi-center prospective studies. They had retrospective studies. They had a variety of other studies that were included. But you can see here that the range is anywhere from about 6% mortality for ICU mortality up to 30% mortality for older adults. And then if you look at in-hospital, that more or less doubles at the lowest range and then goes up to about 40% at the highest range. So a major burden of critical illness. The other thing I'll say is that there's a few studies that have looked at what happens sort of acutely in the hospital, and older adults are sort of overrepresented in those who die very early. So within the first 24 hours of critical illness, 24 to 48 hours. And that becomes even more common if you come in with pre-existing frailty. So certainly a patient population that we should be well aware of and be very thoughtful with when managing. And there's some data out there to say that we just don't treat them exactly the same. They may not get antibiotics as quickly. We may not get source control as quickly. And so sort of understanding why that might happen is important. And then long-term, this is a study out of Finland shown on the right. This has 3-year outcomes for patients who are admitted to an ICU. You see most of the mortality happens within the first month or so, but certainly there's a tail where out to 1 year, you have about 50 to 60% mortality, depending upon the age range. I will note that if you look at different places in the world, so Australia, Germany, and Korea, that mortality may be lower. Some of this likely has to do with what their ICU admits. So for example, Australia and New Zealand, I think, has lower acuity than the rest of the world and has probably more critically ill, quote, critically ill patients. That said, their ICU mortality is still 8.5%, so still well within the range of what I showed in that prior slide. And then you can look at Germany, where likely their longer term outcomes, that 1-year outcome, may have to do with health systems factors that actually improve that longer term mortality for those greater than 90 years old, which is really remarkable that only 35% of those patients are dying out to 1 year. So I think there are some health systems issues that could certainly make things better here. For sepsis, similar story, again, going back to Mark's paper from about 20 years ago now, there's certainly an age-dependent risk of in-hospital mortality for sepsis specifically, more gram-negative rod sepsis in those over 65, and potentially more gram-positive and fungal infections than those under 65, likely due to immune suppression and oncology and HIV-related causes, and no real major change in source of infections. We still see mainly pneumonia and GU sources, so urinary tract infections, but the rest of the causes sort of fall in line. And this is the chart sort of describing that age-related mortality here on the right, where you see that our oldest adults have a case fatality rate of about 25% for in-hospital mortality. That's actually 30-day mortality. And then for ARDS, a group at Vanderbilt looked at this in a relatively large study that showed that for all ARDS, for every decade of life, there's about a 17% increased risk of death. And this seems to be predominantly for direct ARDS, so pneumonia, as well as aspiration, where there's a 30% increased risk of death per decade of life, as shown here on the right. Whereas indirect ARDS may have less of a sort of an age-dependent effect. Why that is, I think, is left to be seen and requires additional sort of preclinical and translational studies to understand that intersection. And then I'll point this out. This was a small editorial that aggregated a few data points for adults admitted with shock specifically. So septic shock, cardiogenic shock, not really differentiating between causes. And they were actually highlighting this 97% mortality at 1 year for those who had circulatory failure. And their point was that we've got room for improvement. We can do better than this, we think, if we are thoughtful and considerate. But I think this suggests that those who are sickest, those who require vasopressors, have that sort of the worst outcomes, and we need to think about how we might do better for these patients in particular. Okay, so that's general sort of in-hospital outcomes. What do people experience? How many people survive? What I'll go into next, and I'll just briefly touch on it, but I think this is probably the burden that you all see in your clinics, is really what are the long-term outcomes of critical illness. And so first I'll talk about survivorship. The first thing that we know is that cognitive impairment and critical illness, and really hospitalizations in general, I think is borne out. There's a double risk of incident dementia in those who survive illness in general. That's a good study from 2010 done at the University of Washington. And then there's a paper by Jack Awashina looking specifically at sepsis, and it was a health systems project that showed a 3-fold increased risk of moderate to severe cognitive impairment in survivors of severe sepsis, especially in older adults. These patients were all above about 70 years old and used claims data and sort of understanding trajectory of cognitive outcomes to argue that this is a major burden for older adults. But I will say that this may not be limited to older adults. So this is a study from the Vanderbilt group again, where they did 3-month and 12-month outcomes for patients using the R-Bands global score. And you see here, it's kind of hard to read, but they're equating it to mild cognitive impairment, traumatic brain injury, and Alzheimer's disease on the far right. And so looking at the levels of what this R-Band score should look like for these different populations. And you see really across age ranges, there is this major impact on cognitive outcomes. And you can imagine that our older adults are probably less resilient than our 45-year-old to sort of adjust for these changes, but this does not solely impact older adults. It probably impacts all comers to the ICU. So I'll just point that out. And similarly for function, if you look at disability, this is work done by Lauren Ferranti at Yale and her group. And there's a couple things I want to point out here. Is first, if you look at patients who come in with minimal disability into the ICU, you see that about 40% of patients will end up with mild to moderate or severe disability after discharge. And that does not account for the worst form of disability, which is death. So about 50% of patients are not going to do well at all, at least in the 3 to 6 months following discharge from the ICU. And similarly, mild to moderate, 40% of them will go on to do worse. And then severe disability, I'll point out that 30% of patients die who have severe disability coming in, and the rest don't get better. They don't improve from there. And so this points out two important things. There's a trajectory after critical illness that people become less functional, but it's also that if you have a sort of a pre-existing poor functional status or some degree of disability, you're at much increased risk of death as well when you're in the ICU. Okay. So this is a really nice summary sheet that was put out by Margaret Herridge in Toronto that really highlights sort of what happens in the ICU that I think you all are well aware of in this room. But you see that orange is the patient themselves, and this was focused on sort of say a 40-year-old adult patient with children. But you have multiple sort of areas of challenge for the entire group of people taking care of this patient, including the patients themselves. You can have tooth injuries, you can have bed sores, you can have functional impairment, I've already pointed out, cognitive impairment, PTSD, mental health disorders for the patient themselves. There's actually an increased mortality in caregivers for those who take care of their post-ICU patients, their loved ones who've gone through the ICU. Children can deal with PTSD and having to deal with the fact that their loved ones almost died or did die. And then for us, I think there's some moral ambiguity sometimes here and some moral distress that can come up. And it can be really difficult to sort of do this on a regular basis, especially as a resident. I think as attending, I've come around to be able to do this without too much moral distress, but still it's there. So this affects everybody, and I think I just want to highlight that and why it's an important thing to study and think about. So in summary, I'll point out I'm also using a lot of Google Gemini photos. So this doesn't totally make sense, but I'll explain what it means in a second. So critical illness in older adults is very common. It remains very invasive, like every patient we take care of in the ICU. In general, it's associated with higher mortality, and life for survivors is just difficult, and that's, I think, what you all see on a regular basis on the outpatient side as well as the inpatient side when they come out of our ICU. But I think one of the things, because there is such a burden, there's opportunity, and that's what this is saying, right? This person is trying to climb out of a valley, so there's opportunity for improvement and to do better than how we do now. At least I hope so, and so that's where sort of I come in. And so my interest is at this sort of weird interface. When I did fellowship here, and so where I started really was the interface of these four things: critical care, which is what I do in my clinical job, but also what I care about in terms of the sort of understanding the pathophysiology of it, and specifically sepsis and ARDS, neurobiology, so what happens to the brain, glycobiology, which I'll explain in a bit and we'll dive into in a few minutes, and then endothelial biology. And what I've ended up doing with my GemStar and over the last 5 years is sort of integrate these four things with aging and sort of understanding aging biology in the context of this really interesting, sort of strange intersection that you probably never thought of. My group, this is sort of a lofty 35-year goal, this is what I focus on. So I kind of focus on all of the things related to critical illness. I've done work in sepsis, ARDS, TBI, cardiac arrest. I've looked at delirium, and that's actually what my K is on and just about to finish this year, but looking at brain dysfunction during critical illness. I'm definitely interested in organ injury and death. Of course, I don't want to ignore that, some studies say that 97% of our older adults are dying, so we should probably work on that and do a better job with that. But then also, I deeply care about post-ICU syndrome. This is really where I start, is trying to get people who do survive to do better in the long run, which I may not be the best person at that, but I can provide hopefully some biological plausibility to some strategies that may help. So the rest of my talk, and I'll explain why I'm talking about sepsis specifically, is because of this. I think if you look at hospitalized patients, hospitalized adults, and you look at cause of death in the hospital, a third of deaths are going to be due to sepsis, according to one single study that actually looked at medical errors. Nonetheless, they took a random sample of 600 patients who died in the hospital, and 35% of those patients will have an immediate cause of death of sepsis. So it's extraordinarily common in the hospital, and I think important for us to understand and do better with. But that said, there's some applicability of this to ARDS, some applicability to potentially other critical illness syndromes, but really my focus is on sepsis in this talk. Okay, so that's really the background. What I want to begin to sort of explain why this is an important intersection, and I think you all understand that and appreciate that, and probably much more than I do as an intensivist. That said, my work, I'm going to talk about two projects. The first one is really the primary focus of what my lab's doing now. And the other one is sort of a side project that I'm really excited about and want to share what we've done so far. So I'll first talk about really the endothelial injury component of this and what I'm focusing on sort of in the immediate future. So the intersection of sepsis and aging from the basic science standpoint is, and really translational science standpoint, is not super well understood. I think plenty of groups have looked at the importance of immune dysregulation, systemic inflammation, coagulation, and the importance of specific comorbidities on outcomes. Things like COPD have an increased risk of poor outcomes. But the one thing I will say is that almost nothing has been done on endothelial injury, and that often goes for all of critical illness, let alone critical illness and sepsis in older adults. And so that's where I come in, is trying to understand this interesting phenomenon that we think is really important for both short and long-term outcomes in critical illness. Okay, so now for the basic science, I'm going to dive into it so I can give you a primer on what I do. So I study these sugars called glycosaminoglycans. Glycosaminoglycans, there's a whole conference on them. There's a Gordon conference that I go to once a year talking to all the glycochemists that do this type of work. But they're long, linear polysaccharides made of these repeating disaccharide subunits. Not so dissimilar from DNA or RNA. They just happen to be somewhat more complex and less understood than DNA and RNA. They can be hundreds of sugars in length, and there are three types that I generally focus on, but there are a few others that I'm not mentioning here today. And the one I'll talk about primarily today is heparin sulfate. Heparin sulfate should sound familiar because we all use heparin in our daily lives, and heparin is just a form of heparan sulfate that has a sort of certain pattern of a certain structure that gives it its anticoagulant effect. But so heparin sulfate is one of these, and I'll talk more about that. And one of the things that my mentor's lab and my lab have been able to do is actually look at these sugars and do effectively glycomics, if I want to call it that, by using mass spectrometry to measure these in blood and brain and urine, whatever biospecimen we want to look at, which is sort of a unique component of what we do in our lab. The analyses are sort of rudimentary compared to, say, proteomics or other forms of sort of these multi-omics, but we get some interesting data from these results. This is heparin sulfate. So again, long polysaccharide, sometimes up to 100 sugars in length, but typically anywhere from 5 to 40 sugars in length. You see that there are these S's. These are sulfation side chain or sulfation patterning across that chain. These are typically attached to a protein, and this is called the heparin sulfate proteoglycan. So this whole structure is that. So they're always sort of generally attached to a protein, and they're attached to endothelial cells, and that's why I care about them and why I'm interested in them. And so we can do an electron microscopy. This is not my work. This is this is shown a ton in our glycobiology talks, but this is the endothelial glycocalyx, which almost looks like a furry layer on the luminal side of endothelial capillaries. And it juts out into the vascular lumen, and you can imagine what this does, but I'll dive into it a bit more. This was an electron micrograph done by some colleagues in the University of Nottingham, and I now show this because he's always mad that we only show this picture. So anyway, showing this one too, which is another representation of what the glycocalyx can look like. And there's some really nice trend like TEM, so those three-dimensional images that show this really nicely, both in sepsis and not sepsis. So I'll talk about why this matters in just a moment. And just to reiterate, and I'll come back to this, a little bit less important in this moment, but it has this sulfation patterning, which becomes really important. In fact, heparin sulfate is the most negatively charged molecule in our body. So, and that has importance because of this. So if we look at a heparin sulfate proteoglycan, it has this negative charge patterning. And what that does is it allows it to specifically interact with proteins and often acts as like a docking member of a protein receptor interaction. So that's what this most commonly does. But effectively, heparin sulfate, they have pleiotropic effects on biology because they interact with so many proteins. In fact, if you look at heparin, it might interact with 500 proteins. And the question is sort of why and how and what happens as that patterning changes. After sepsis, why this is interesting is that the glycocalyx is degraded, or during sepsis, we know that this heparin sulfate chain gets shed into the bloodstream, and actually the core protein does as well. And so I became fairly interested in why degradation happens. And so it turns out that this negative charge also confers, or dependent upon how the negative charge is distributed, degradation may happen differentially. And so I'll come back to that. And that basically there's enough heterogeneity in this molecule that may change with age or different factors that could predispose it to degradation. So that's really what this talk is going to be. This part of the talk will be about. The glycocalyx matters for a variety of reasons, and there are probably more reasons that it is important as we move forward in this field, but we know that it has an impact on limiting vascular permeability. So if we degrade it, vascular permeability increases. You get more leak, if you will, capillary leak if you degrade the glycocalyx. It restricts circulating cells to the vascular lumen instead of sort of extravasating into the lungs or other organs. It modulates inflammation and coagulation. So if you get rid of your heparin sulfate on the endothelial cell, coagulation may occur in that vascular bed. And then it's also a sensor of endovascular forces, so has sort of interacts with intracellular machinery to transduce basically what sort of stress is being seen within a cell. The other thing, and this was what I did in my fellowship, is that we actually found, interestingly, that the sugar fragments themselves may cross the blood-brain barrier, specifically in the hippocampus, and affect memory in survivors of sepsis. And this is really where my work started. This is a quick 10,000-foot view of what we did, but this was published back in 2019. And again, has that sort of really specific protein interaction. What we know about the glycocalyx in aging is that it's probably thinner with aging and sort of a healthy adult. The basic composition changes, although those compositional changes are a bit hand-wavy and not entirely understood. And then there was one group that has hypothesized that age-related endothelial glycocalyx deterioration is an initiating factor in vascular dysfunction and advanced age. And what that's trying to lay out is that for chronic diseases, that endothelial dysfunction, endothelial glycocalyx sort of diminishment may actually have an impact on things like hypertension, stroke risk, things like that. And so this is what this group believes, is that chronic vascular diseases may sort of be involved. The endothelial glycocalyx may be involved in that. And I'm really interested in the acute disease, right? I'm interested in sepsis and other forms of shock. Okay. So again, I'll remind you, the endothelial glycocalyx does this during sepsis. And what we don't know is what happens with aging. How does aging affect the septic endothelial glycocalyx? And really, up to this point, we do not know. There's no work that's been done in this space. So interim summary number 2, I talked about number 1, which was about the burden of critical illness in older adults. But what we know is that sepsis is common, has a disproportionate impact on older adults. The glycocalyx is an important homeostatic endovascular layer that I can study using some cool tools. Endothelial glycocalyx degradation has important pathologic impacts that have importance for outcomes, both short-term and long-term. And the intersection between aging, sepsis, and endothelial glycocalyx degradation is completely unexplored. Okay. All right, so again, I'll point out that this is what we can do. We can measure in plasma samples these heparin sulfate fragments, as well as syndecan-1, which is the protein that most of these heparin sulfate molecules are attached to. And so we can look at the trajectory of glycocalyx degradation in patients. And so we did this a few years ago now, and this was sort of a secondary analysis of this data set where we had about 100 patient samples. We have control as well as sepsis patients, and this was sepsis and septic shock, so a continuum of disease severity. And you'll see here that compared to age-matched controls, patients with sepsis less than 50 did not have a significant increase in glycocalyx degradation early on in sepsis. Similarly, 50 to 64, although pushing towards the limit of significance, had no statistically significant increase in glycocalyx fragments in their blood, whereas those greater than 64 had the most profound increase, so suggesting that there's some age-related effect on glycocalyx degradation. Just at a really simple, I'm a basic translational scientist, so forgive me, but multivariate analysis looking at sort of what factors impacted heparan sulfate shedding, and age did seem to have an impact, independent of SOFA score and gender being the other factors that we controlled for. And this was a relatively small cohort, so it can only control for so much. We can do this in mice, which is where I come in. So we can inject, most of our experiments have been done with LPS because it's reproducible. We kind of know how to titrate it to mortality. And so we did a moderate dose of LPS that is about 20% mortality in an older animal, tends to be 0% mortality in a young mouse. And we looked at very young mice, which is between 8 and 16 weeks old. So, adolescent to early adult, compared to 20 to 24-month-old mice. And what you see here is that both heparan sulfate, which is again that sugar molecule, as well as syndicin-1, which is that core protein, only increase in the older animals after LPS at this dose. So suggesting that the glycocalyx sort of is more susceptible to degradation in our older animals, similar to our humans. That said, with a higher dose of LPS, I will say that we do see glycocalyx degradation if we give sort of a bigger hit to these animals. One specific pathway that I'm interested in is heparinase 1 upregulation. This was really the foundation of my mentor's work, is that he actually found that TNF-alpha related heparinase 1 upregulation, or really activation, leads to the sort of first pass of glycocalyx degradation, and showed that if you knock out heparinase, you can protect from this. But that's the molecule that I'm particularly interested in because it directly degrades that heparan sulfate chain that I've talked about. And so this is what it does. Heparinase is activated within the cell and gets spit out, and then it can degrade that glycocalyx as shown here. And then from there, the thought is that MMPs and other molecules can get in and really degrade that core protein. And it's sort of this cascade of effects for glycocalyx degradation. Okay. So the first thing we did was this was with Vanderbilt again, where we looked at heparinase activity. So this heparinase 1 activity across the age spectrum. And these were all patients who had sepsis. And what they found was that there was no increase in heparinase 1 with age. So we couldn't just blame this simply on more heparinase upregulation in our older adults, and that was the reason for more degradation. So that was not the simple answer, even though that would have been the sort of straightforward and easy to investigate mechanism. So endothelial or interim summary number 2 is that glycocalyx collapse is known to be initiated by heparinase 1 that cleaves heparan sulfate, which we can measure in the blood. There seems to be more endothelial-derived heparan sulfate in sepsis patients that are older during sepsis, and we can also replicate that in an animal model. But there are no age-related increases in heparinase activity during sepsis based upon our studies to date. So that brings me to, again, this basic science, translational science hypothesis, which is that the glycocalyx may actually be more susceptible to heparinase 1 rather than it just be due to more heparinase upregulation during sepsis. And so we used a heparinase analog. There's a bacterial heparinase that acts a lot like the human heparinase. It's called heparinase 3. And I don't know why this shows up like this, but you get the point. So we injected IV heparinase 1 analog, so that molecule that degrades the glycocalyx. We collected plasma 1 hour later, and then we measured those heparan sulfate levels. And what you see is that our young control animals, there's a level about 500 nanogram per mil heparan sulfate circulating. The young who received heparinase had maybe a 10% bump, but it was not significant. And then if you look at our aged animals injected with heparinase 3, you see this sort of 6 to 10-fold increase in heparan sulfate, which looks a heck of a lot like our sepsis model. So it suggests that perhaps the glycocalyx is in fact more susceptible to degradation. So again, I'll remind you, and I showed you this earlier for a reason, just to reinforce it, is that the patterning of this molecule is really important for the ability of heparinase and other degradases to degrade it. And so again, I'll dive a little deeper into the biology of this and the chemistry of this. But we know that if you have this NS2S6S enriched heparan sulfate, it tends to be pretty resistant to heparinase 1. So if you have trisulfated heparan sulfate, heparinase does not degrade it very well. If you remove a 6-O sulfate and you have this NS2S enriched heparan sulfate, it seems to be, it's really sort of the most susceptible to heparinase 1. So if you change the patterning of this heparin sulfate sugar chain, it actually changes its ability to be degraded by that important molecule that I've mentioned. So what we did is so just highlighting that again, NS2S is more susceptible to degradation than NS2S6S. So I'll explain how we looked at this. So we basically wanted to understand, what does the glycocalyx look like of an older adult? So that was our next step. We used a similar experiment. So we used a mix of heparinases. So there are multiple, when we actually measure heparin sulfate levels using mass spectrometry, we actually have to degrade the molecule completely. And to do that, we use heparinase 1 and 3. It just basically is a blunt instrument to cut it into its individual disaccharide components. And that's how we do that analysis. But we can give those same drugs to an animal. So we give heparinase 1 and heparinase 3, or these same molecules, these same enzymes. We can, again, collect plasma, and we can look at the constituent heparin sulfate that's shed using mass spectrometry that's quantitative. And what we found is, first off, the glycocalyx degraded. And then if you look at the absolute values, they're like 30 to 50-fold increases compared to that 5 to 6-fold that I showed with the heparinase 3 alone. But you see that there's this shift towards NS2S compared to trisulfated. So it seems to be a shift towards this more susceptible form of heparin sulfate in our older animals. Why might this occur? So it turns out that there's a lot of machinery to make these molecules. There's a little less that changes it once it's been put on the cell surface. And there are two really interesting molecules called sulfatase 1 and 2 that are the only molecules that modify the glycocalyx once it's on the cell surface. So there's no more change other than sulfatase 1 and sulfatase 2 can modify it. And convenient for me, what it does is it cuts off the 6-sulfation. So it makes that trisulfate become that disulfated, that NS2S, which really does make it more susceptible to heparinase. And in fact, it's more complicated than this when we talk to the chemists, that sulf probably also does other things, hand wavy, that chemists can explain, but I cannot, to facilitate heparinase degradation. So there's two candidate proteins that could be involved in this, sulf1 or sulf2. And again, it clips off that 6-O-sulfation after the heparin sulfate is on the cell surface. And so we looked at this activity of these sulfatases. We can't look at them individually because they have the same impact, but we can look at whether blood of young animals or old animals without any treatment with that heparinase can degrade heparin, and that's what we did in this experiment. And we find that there's an increased sulf1 or sulf2 activity using this special assay that was developed by one of my collaborators in Grenoble. And we did the same thing with a group in North Carolina using a very specialized assay that basically says, and don't worry about the chemistry, that an increase in this 3S concentration suggests an increase in sulf activity. So we've now sort of combined, we've got an idea of why that we see more disulfated instead of trisulfated in our older animals. And so age seems to be increasing this 6-O cutting activity. And we don't know whether it's sulf1 or sulf2, but fortunately for us, a group has done this about 10 years ago now. Rado Goldman is at Georgetown University and studies these sulfatases. There's a few people who do this in the world, but he had done this experiment where he developed an ELISA to look for sulf2 levels, and he found a linear association between sulf2 and age. So suggesting sulf2 is likely the sulfatase involved in this process. And something we'd like to do with him in the future is sort of confirm this finding in a larger cohort. Interestingly, we also see this in cirrhosis, so may have a similar impact in cirrhosis, although we haven't looked at that specifically. And then I'll finish with, we know where sulfs come from. So this glycocalyx modifying enzyme, sulf1 tends to come from endothelial cells, which we've studied in our lab before and have looked at sort of post-septic remodeling of this glycocalyx. But we know sulf2 comes from monocytes. And so I went back to a single-cell RNA sequencing data set that compared younger versus older adults with that were healthy. These were just healthy controls, and included a group with frailty as well. But we looked at the monocyte population and looked at sulf2 expression and activity. And we saw about a 2.5 to 3-fold increase in sulf2 in the monocytes of older patients compared to younger. So suggesting that this could be the source. And this is all sort of the preliminary data for a grant to test this hypothesis. But these are our interim conclusions, and sort of where we're taking this is that the glycocalyx is more susceptible to degradation in older patients. There seems to be no difference in the heparinase 1 activity in younger versus older patients that are affected by sepsis. There's this shift to the non-degradable to degradable form of heparin sulfate in the glycocalyx of older animals and potentially older humans. And this age-related increase may be related to monocyte-derived sulfatase too. And so I have a grant that's sort of pending right now to look at this in more detail and understand the impacts on kidney injury, long-term outcomes, including cognitive impairment, as well as mortality. And so that's where this all goes, is trying to determine whether this axis is important for outcomes. All right, so now something completely different, has nothing to do with endothelial biology, which is the impact of sepsis on hearing, which I think is really fascinating. I don't know if it's going to bear out, but this actually all came about because I was doing a study in my sort of outcomes and delirium work where I was trying to do hearing in mice. And I was just having the hardest time making this work. It's this experiment called pre-pulse inhibition, where I sort of blast a sound at a mouse, and then I prepare the mouse with a blasted sound, a lower sound, and I just couldn't get it to work. And it turns out that there's this guy, Akam Klug, who is now right down the hall from me, who's an auditory physiologist. He does a very specialized pre-pulse inhibition. I was like, I should talk to him because he is interested in hearing, and I can't get this thing to work, and maybe he has a better setup. So that's how this all came about. But it's kind of developed some legs of its own. So this is likely known to you all, but the Lancet Commission put out this report back in 2020 looking at potentially modifiable risk factors for dementia. And I think that these are not coming through very well, but you'll see that actually they call it the population attributable fraction of dementia risk in middle age is hearing loss. Hearing loss is sort of the most impactful factor in those who are between the ages of 45 to 65, accounting for about 10% of the risk of dementia risk moving forward. And so suggests there's an interesting continuum between cognitive impairment and hearing loss. And you all likely know more about this than I do, but this is my really simple reading of the literature. It's also been shown, and this could be confounded, but nonetheless, it's interesting. So Lauren Ferrante's group looked at factors that predicted poor outcomes in survivors of critical illness. And actually, in the lower number here is suggestive of a worse outcome, so less likely to be functionally independent when they leave the hospital. And actually, the most impactful factor here is severe hearing impairment. So kind of interesting that hearing impairment is a big, it's no good for our critically ill patients as well as I think patients in general, and something that's probably an under-recognized sort of epidemic. Here is importance of auditory dysfunction in older adults with delirium. So bringing in that idea that delirium has some mediating effect on these longer term outcomes, which I haven't talked about in any detail. But hearing impairment is a really important factor in risk of delirium, which I think you all know, and that's why we try to get hearing aids in and correct it if we can. And if you look at hearing and visual impairment, the odds ratio is really similar. The increased risk is similar between hearing impairment and hearing and visual impairment combined. Suggests to me maybe hearing impairment might be a little bit more important. That said, they're probably both important. And so making sort of a somewhat complex thing somewhat simple is I'm wondering actually whether sepsis could lead to hearing loss, which I'll show you, whether hearing loss could lead to delirium acutely, and then whether there may be some sort of complex mediation here between hearing loss and these longer term outcomes. By no means is this final, but I think it's an interesting hypothesis. So there's some really bad studies that have looked at this. And one is looking at really a prospective cohort where there was pre and post data for auditory function, and they found that about 20% of patients ended up with an increase in 10 decibels of their auditory threshold, meaning they had lost their hearing. Greater than 10% auditory threshold is suggestive of some auditory dysfunction. And that was after ICU stay specifically, and not focused on sepsis in particular, and they didn't divide by sepsis, but suggests that there could be some legs there that ICU stays may lead to worse hearing impairment. The other one, which is ridiculous, is that there's a 50-fold increased risk of hearing impairment after sepsis in sort of 18 to 65-year-olds using a huge registry in Taiwan. Very unclear what this means. That said, it's a really impressive number. And this is the chart that they show, is that there's a huge increased risk over 10 years after an admission for sepsis. So some, really kind of poor data to suggest that this could be an interesting axis. But then it turns out that there's some preclinical biological plausibility to this, where a group in Austria has actually looked at this in some detail and has published a couple papers on it, that if we do our sepsis model, cecal ligation and puncture, you actually can end up with a notopathy, where you end up with sort of hardening or sclerosis of your inner ear after a systemic inflammatory response to infection. And so there is some data that's actually quite high quality to suggest that there is an axis here, at least in mice. And so decided to look at this with Achim Klug, who's the guy here on the left, and then Tim Lay, who's an electrical engineer, who they've worked together for quite some time, but I kind of joined the group. And we can assess hearing in rodent models using this method called auditory brainstem responses, which you can also do in humans, which is quite convenient for me. And what we get from this, we get two primary pieces of data. We get auditory threshold, as well as the sort of inner workings of the ear and brainstem. So we get both a threshold, which is basically where we can see a response in these animals. We lightly anesthetize these animals to be able to do these experiments. But then we can also look at this complex 5-wave response that suggests where the pathology might be happening. So is it the auditory nerve or somewhere deeper in the brain? And this is sort of Achim's space that he really understands well. So we did this in mice. And interestingly, we found that there's sort of an acute increase in the auditory threshold in mice injected with LPS. So this is our, our workhorse model. We're at 72 hours. We had about a 10 decibel increase in our auditory threshold in very young animals. These are 8 to 12-week-old animals. So these adolescent type animals. And when you divide that by survivors, doesn't seem to be an impact in survivors, but non-survivors, there is a statistically significant increase. So suggests that maybe this has some importance for outcomes. And, when I look at these data, I'm wondering if, had we supported these survivors, would these be the ones that would go on to end up with sort of longer-term auditory dysfunction? The other thing that you probably will never hear anywhere else is that you cannot do age-related studies of hearing in mice, because black 6 mice go deaf after 6 months. There's a SNP in there that apparently leads to deafness starting at about 6 months. So to be able to look at older age, you end up having to use gerbils or some other animal. But gerbils have an auditory system that's very similar to humans. And so we did this in just a small initial cohort of gerbils. And a little different, but similar, we saw something interesting, which again, preliminary, but nonetheless interesting, is that by 3 days in this, we had to use a lower dose of LPS because these are USDA regulated animals and we really didn't want them to die if we could avoid it. At 3 days, we did see about a 10 decibel increase in their auditory threshold, so consistent with mice. And then at 28 days, you'll see that there's some drop off because there was some mortality here. There was a 10 to 20-fold increase in auditory threshold in these survivors. And again, these are sort of young to middle-aged gerbils, not the aged gerbils. So suggesting we can test this in a rodent model. Recently, we tried to do this with very old gerbils and did not go great because gerbils are very susceptible to LPS, it turns out. So lots of interesting biological heterogeneity by species, but we're working on that as we speak. So for this project, this is supported by the Team Collaborate grant. We started with just the team formation grant about 2 years ago now, and then have started to dive into this with the full Collaborate grant. And so our intention is to look at young mice and gerbils and sort of round out that initial data set. Certainly have to do more ends with the young gerbils. And then attempt to do this in aged gerbils to see if there may be an age-dependent effect as well, which is difficult because our vets do not like sick gerbils. So we're working on that, trying to work out that piece. And then the other thing that I think is actually more fun, trying to do a small translational study, which I have now IRB approval for, but have to just get all my ducks in a row to do this. And the idea is that we'll have 3. This is just going to be a pilot feasibility study. It's been done by Lauren Ferranti before, although using different methods that I'm planning to use. She said it was very hard, so I don't know if I'll be able to do it. Nonetheless, I'm going to try. So our intention is to do HHI-A screening, so hearing, it's an auditory index that basically allows us to determine, or at least guess, whether somebody had pre-existing hearing loss before their hospital stay. So we'll talk to either their loved ones or themselves about it. If they do not have pre-existing hearing loss based upon the survey, we would do audiograms, auditory brainstem responses, and otoacoustic emissions, which are just 3 really straightforward things you can do without any patient participation, and do that throughout the course of their hospital stay. So do it at sort of day 0, within a couple of days of their admission for sepsis. Do it later at 7 days or near time of discharge. And then ideally bring them back to the clinic, but at least call them about the HHIA on the back end to see if things have changed. And we'll have a sepsis group, a non-sepsis group, and then a control group in this study, just as a pilot feasibility to see if we can do it. The other really cool thing about this is we can do it in critically ill anesthetized patients. So we can do this with propofol on board, we can do it with midazolam on board, and we can still get ABRs. So there's something here. We may be able to look at hearing actually even in our critically ill patients. So we'd like to do that in the future, but that would be sort of a next step in this project. So giving you a lot of different things. I've given you hopefully a reasonable overview of what critical illness looks like for our older adults, given you my sort of primary hypothesis that I'm testing and hoping to continue to work on in the coming years. And then this little tease of a fun project that I hope picks up some legs and working on over the next year or so. Lots of people that I've worked with to do this work. And just really glad that I could be here today and talk to y'all. I'm excited for some feedback, and if anybody has ideas, would love to hear them. So I'll leave it there. In case you feel like them, feel free to put on some sunglasses and go to sleep.
You, as people are waiting or thinking of your question, I'll start off. So if someone develops delirium after critical illness and a hospital stay, should we be looking for potentially incident new hearing loss that's there?
Yeah, I think it's a great idea to do. Or it was maybe unrecognized. I mean, I think this is the real challenge in the hearing loss component of this work is, did they have pre-existing hearing loss? I think it's the same thing with the decision about, is incident dementia real after hospitalization, or is it that they had pre-existing dementia or a predisposition to it? I think it's an interesting idea, and we should probably assess. It's probably difficult to get an audiogram or an ABR in the hospital, but maybe we can make that worthwhile. And if nothing else, even if it was pre-existing hearing loss, it's probably good to know for people to be able to move forward and have good care. So I think a good thing to do no matter what, even if we don't find that it's mechanistically linked to sepsis.
Hi, thanks. Wonderful talk. One, thank you for studying old mice that are actually old. And I'll ask this question because Carrie Moreau's not here, but have you looked at sex differences in any of these outcomes in your mice? And that'll be my first question.
Yeah, so the sex differences, so in the human study, at least we didn't see an independent association between glycocalyx degradation and sex. I will say that endothelial is a little different than epithelial. So I study endothelial dysfunction. My mentor has sort of moved to epithelial dysfunction. So epithelium is the alveolar side, what happens in the airspace, not so much what happens in the circulation. That becomes somewhat strategic because lung injury we can study that in preclinical models, whereas endothelial injury, we cannot study in sepsis preclinical models without clever ways of doing it, because NIGMS won't let us study sepsis in preclinical models. But there are absolutely sex differences in alveolar epithelial glycocalyx degradation, where females tend to have, and I might be conflating, I might be mixing them up, but I think males have a higher risk of epithelial degradation, whereas females have almost no risk of epithelial glycocalyx degradation, which may have an impact on their ARDS outcomes. In our animal models, we always include both males and females, and we have not seen necessarily a sex-dependent effect, and we do look at those separately. So some of like if you look at the mice, I've done this red and black, but I'll usually have sort of open circles or closed circles here to see if there is a sex effect, and I think it's a very reasonable question. In general, I think in our preclinical models, we see that females tend to be more resilient to infection than our males for whatever reason. And so, yeah, there are likely to be sex differences, and I think a whole nother sort of can of worms to open up to talk about the sex variability of these outcomes.
Thank you.
Yeah.
I'll hold my.
Thank you. Do you mind going back to that mechanistic image you had where the enzymes are released from the endothelial cells? My question is, in aging, does the glycocalyx release an signaling factor for the endothelial cells to release the enzyme? And then my second question is, in young, healthy mice, that mechanism, is that released in response to just damaged glycocalyx then repairing, or back with the aging, is that dysregulated? So now you have a shift to these enzymes being released more, even though the glycocalyx may be okay.
Yeah. Yeah, I think it's a great question. I think this has been somewhat glossed over, I think, and it's hard to get funded to do the mechanistic work in this space where you're looking at the transition from proheparinase to heparinase. I think what I understand of this general pathway is that proheparinase is floating around. There's always proheparinase around. That's activated by a cathepsin. Cathepsins are these proteases that cleave off some of a protein. Cathepsin L is thought to be implicated in this, although I've looked at cathepsin L in the blood of at least young animals, and there's not an increase in sepsis, so it's a little unclear. It might be intracellular cathepsin L causing that degradation. But in general, this is thought to be upstream of this is TNF alpha. And so I think there's a very reasonable question as to whether TNF alpha is higher in older adults compared to younger who come in with sepsis. In our mouse model, that's not the case. There seems to be a prolonged TNF alpha elevation in our aged animals, but not necessarily a different peak, if that makes sense. So upstream of the sort of proheparinase to heparinase, I don't think there's any major difference, although maybe there's sort of a longer insult that could be explanatory for why this happens. The other thing that does happen is that once heparinase has degraded the glycocalyx, we actually know that it activates the endothelial cell. And so I do think that there's something interesting there where once you have heparan sulfate degradation, it leads to the way that it's been described as a forest with a tree without its leaves and branches. So the branches go away and all of a sudden the protein has more mobility. And this is a group at UAB that's done this work. That mobility leads to some intracellular signaling that leads to an increase in Ang2, angiopoietin-2, that likely has sort of endothelial activating effects. And so this pathway is, I think, important. It's hard to do in a whole animal. I think it's possible to do in cell culture assays, but I think it also requires flow. So you can't do these experiments, I think, well without flow. So that said, that's a great question. I don't know the answer to it, but the intricacies of this pathway, I think, are important to work out, but it requires some people who can do good cell culture technique, and I think cell culture with flow, and then using aged versus young. And I think then you can really, dig into that pathway and understand sort of what impacts are most important. That's kind of beyond me. I'm a whole animal person.
Thank you.
Okay, I'm going to move far away from this because it's a little overwhelming to me. So I guess I think back to the beginning where you were talking about like how like the incredibly high mortality for older adults, and you said your focus was on how we help them survive. And I think about the opposite of how we identify those that we aren't going to do all this to, and have earlier goals of care conversations. In thinking about this, do you think this like the endothelial dysfunction in the heparinase, do you think this could be used as a predictor of who maybe won't do well and could maybe help with prognostication and maybe earlier goals of care conversations, or thinking about who we don't offer some of the interventions to?
Yeah, for sure. I think so my mentor did sort of the biggest study in this as of about 6 months ago, or finished, and it was published. So we know that heparan sulfate levels are they're not perfectly predictive of mortality risk, but if you have sort of the highest quartile of heparan sulfate, you're at about 40 to 50% mortality risk. And so, yeah, it could certainly be a prognosticator. I mean, my hope is that, and this is, I think, the hope in critical illness in general, is that we take sepsis and we're like, everybody's sepsis. And we should treat them all the same. And they should all get the same fluids. They should all get some antibiotics that cover what they have. And that's all we can really do. But I think the hope is that even at that very early time points, that 12-hour time point, people die in 24 hours. Maybe we can find some biological heterogeneity that suggests maybe the 70-year-old has some biology that we can actually leverage. And that's sort of the thoughtful way of doing critical illness sort of upstream of those goals of care discussions. But yeah, I think heparan sulfate could be used. I think there are other ones though too. I think SOFA scores are pretty good. And undoubtedly, I engage all my patients that come in with severe critical illness, and I'm really clear about what I anticipate the outcomes will be. I think comorbidities have a huge impact on that. I mean, I was talking this past week about a patient who was 78 with new AML. And I was very clear with the family that I don't think this is going to go super well as an intensivist who's taking care of this patient before they'd even started chemotherapy. And so, yeah, I think it's absolutely important to go upstream of this. Hard for me to do that in a mouse, though. So that said, I think we could find like enrichment strategies within a mouse to say, can we look at the heterogeneity within a mouse blood to figure out like the 10% that die, what's happening? Like what is the biology of that? Yeah, if you could avoid that, for sure.
I'll try to keep. I have a lot of questions, Joe. Thanks so much for a great talk. I know we've chatted about our shared interest in endothelial cell biology, but I first just wanted to give you a shout out. I think it's awesome to see this bridge that you've created between clinical practice and basic to translational science. I think it's really inspiring. So one question is technical, the other is a little bit more conceptual. I'll start with technical. So we were doing a study in cancer survivors trying to understand how their cardiovascular health is affected by the chemotherapy that they received. Do you think months, years past chemotherapy treatment, can we detect these degradation byproducts in blood samples to try to infer if glycocalyx degradation might be implicated in the cardiovascular disease risk in these populations?
My opinion of that, and that's a very common question, and like involved in a pediatric study that one of the PIs wanted to ask that same question, like does prolonged sort of glycocalyx degradation or glycocalyx damage matter? I don't think it does. I think there's a sort of 1 week turnaround of this process. So I think after a week, we still see the levels being high, but I anticipate at day 14, day 28, it's all back to normal. I think the question is whether that upfront hit is dependent upon sort of an endothelial degradation process. So having those acute time point plasma samples, for example, would be interesting to sort of determine whether that's when they've split. I do think that mice and preclinical models allow us to interrogate this a little bit more, where we can look for the changes that happen in the glycocalyx using the group at Utah did all of that aging related stuff where you can look at the glycocalyx using funny microscopy under the tongue or on the skin. And so maybe there's a thinness to it. Maybe there's a change in constituency. Those are really hard experiments to do in humans. You have the cool technique of scraping out some endothelium and maybe getting some of those data. And you can use really small amounts of sample to get those data. So you may see some fundamental shifts in what's in there, but I think it's a little harder to say that it's ongoing degradation, if that makes sense.
Yeah, got it. Thanks. We're doing some acute studies, so probably follow up with you on that. Maybe we can measure some things in the blood. Second, more conceptually. So I think you had a figure early on that pointed to cardiovascular disease sort of feeding into sepsis.
Yeah,
if I recall.
Yeah, kind of this aging biology.
Yeah.
Figure.
Yeah, so that I would, and I've thought about sepsis. Say if a younger patient undergoes sepsis, but they survive sepsis, they're now at increased risk for developing cardiovascular disease. So perhaps thinking about it, sepsis pointing to cardiovascular disease. I'm just curious to get your thoughts on what you think the role glycocalyx degradation plays in that elevated cardiovascular disease risk into sepsis survivorship.
I think another great question. There's a lot of work being done in cardiovascular health for sure in this space. There's actually that monocyte SALT2 source comes from work in post-MI care. And we know that glycocalyx degradation and glycocalyx remodeling has an impact on how people do after an MI, for example. And if you sort of improve glycocalyx degradation early, glycocalyx resilience early on and direct cardiovascular hit, it can improve scar and sort of degree of injury. So I wouldn't put it past the world to suggest that the glycocalyx has an impact on cardiovascular outcomes as well. Again, I think similar problem where you have to figure out how does the acute hit impact function long term, but I think if you look 6 months later, it's difficult to sort of tease out without that upfront data.
Well, thanks so much. Appreciate it.
Yeah.
So we will now have our
Wait,
like
that's
10-minute
here.
break before we start
That's-
division meeting.
Okay.
We've got
Thank you. That's fun.
MCOA as the pillar presenting today
Good luck with
and a
it.
number of other things related to philanthropy and other topics. So please come back in like 10 minutes and we'll start our division meeting.
And it's an actual game. So it's all themed. That's fun. Our wellness directors.
It's amazing.
coordinating it. It's pretty fun. I'm the forest. I'm the poison eggnog.
That's so fun. That's good.
I know there's not very many people. There's gonna be like 3 people here. Not very many people today. We're